by the International Society for Development and Psychobiology. She's going to talk to us about brain imaging in neurodevelopment of self-regulation. Please welcome her.
Thank you so much. Thank you for being here and bearing with us as we try to just get in here. So we've all already had our self-regulatory challenge for today. Wait a few minutes. So I want to begin with a relatively simple question today, which is, who here has ever experienced an emotion? All right, we have a few who are a little confused about the question, so we can address that later. But for the most part, most of you have experienced an emotion. How many of you have experienced an emotion today? I have a toddler. I'm 7 months pregnant. I experience emotions like every second of the day. So this is something that I think we can all agree on, that emotions are a part of life. They are what gives our lives meaning. They're what motivate us to pursue challenges. They are the thing that often gets us out of bed in the morning. So sometimes emotions get a bad rap as though they're just these kind of trivial, silly things, but they really have a lot of significance. At the same time, what could also be problematic, is almost as problematic as not having emotions, is perhaps having too many, or having emotions that are not adaptive. So we don't want to go through life being puppets on a string with our emotions pulling a string. Indeed, it's important that we learn how to adaptively regulate our emotions, so as to preserve our relationships, to hold down our jobs, to figure out how to get off of social media when it's no longer helpful, to keep our New Year's resolutions. There are many reasons that learning how to regulate emotions are a really big deal. Just think about all the people you know who are really smart, who have done really dumb things. When you consider this, it's almost always not because they didn't understand that something was a bad idea. It's usually because of some kind of a self-regulatory failure. We see this with politicians, we see this with artists, we see this occasionally with academics. All kinds of folks who are super smart but do dumb things because of problems with self-regulation. So this is a non-trivial skill, this ability to keep our emotions and our impulses in check. And what my lab really strives to understand is how we build this skill over the course of development. What are the fundamental neurodevelopmental building blocks of self-regulation? And what is it that gives rise to the rich diversity of individual differences related to self-regulation? And so, in trying to address this question, before diving into the approach that we typically take, I want to briefly motivate the rationale behind understanding the development of self-regulation. So it's not just, I think that studying kids is important because they're cute, or because they're bad at self-regulation, or anything as simple as that. Really, there are some extremely important scientific reasons for understanding the developmental underpinnings of self-regulation. The first of which is that children and adolescents have good reasons to feel a lot of emotion. So I'm 35. My daily life is not that variable. I've been married to the same person for several years. I've had the same job for several years. I've had the same best friend for like 20 years. Not much is changing from day to day. When you take the average life of a 13-year-old, nothing could be further from that. Think about it. Each day, their social sphere is changing quite dramatically. The transition just from elementary to middle school is enormous with regards to the self-regulatory demands being placed on an individual. Moving from having one teacher giving you the structured educational context to switching to having to navigate yourself from class to class, having to figure out how to allot time to homework, having to resist the temptations of watching a million YouTube videos and get your stuff done. These are huge demands that are being placed on children and adolescents, and at the same time, they're trying to face these demands with brain systems that are very much under construction, particularly when it comes to brain systems that support self-regulation. And it's this really interesting paradox of childhood and adolescence being characterized by high demands on self-regulation and circuitry that's still in process that has led many to conclude that this period, particularly adolescence, is something of a make or break time when it comes to self-regulatory processes. So what I mean by this is that, as I'll be illustrating today, for most individuals, self-regulatory abilities improve dramatically during adolescence. But for some individuals, this is the beginning of what could be a lifelong struggle with self-regulation. And it's perhaps no accident when you consider this, that so many mental health problems that are characterized by difficulties with self-regulation have their first emergence during the adolescent period. So this is, even though when you look across different kinds of mental health disorders that are really diverse in their manifestations, a common core that you see across so many of them that first have their onset in adolescence are difficulties with emotion regulation. Whether that be looking at difficulties with regulating negative affects, such as in the context of anxiety, or when you think about difficulties with experiencing reward, perhaps in the context of depression, or when you look at the desire to pursue perhaps unhealthy rewards in the context of substance use. There's a common core here to difficulties with self-regulation. And so for this reason, my lab is really, really preoccupied by this question of how do we develop self-regulation, not only for wanting to address this as a basic scientific question, to really create something of a growth chart for what self-regulation should look like, but also because I think that this question has tremendous translational potential. So today I'm going to be giving you a talk in two parts. First, I'm going to be presenting some basic findings that I've obtained over the years, looking at the development of self-regulation with regards to the brain and behavior. And then in the second part, I'll be presenting some more new work. This is really hot off the presses, not even on the presses yet, some of it, so bear with me. But this is going to be looking at neural specialization as a core ingredient of self-regulation development. So we're going to have kind of something for everyone, hopefully, here today. So a big challenge for folks like myself who are interested in studying emotion and self-regulation is how do we try to study these processes in a lab, much less in an MRI scanner. We're really good at feeling emotions in everyday life, as we've already discussed today, but it's quite challenging to figure out how to do this when someone is alone in a laboratory context and we have certain ethical considerations we have to deal with, right? So before I actually introduce you to the paradigm that we've most commonly used, I want to put forward two caveats. The first is that the paradigm that we use to elicit emotion and to actually model self-regulation is probably much more subtle than what people experience in everyday life. So when you consider that we're probably eliciting much milder emotional experiences than what people actually experience in everyday life, the fact that we see quite pronounced age-related change is striking. This is the most conservative test, in a sense, that we can get at. We could imagine, therefore, that in real life, probably age-related differences are even greater than anything I'm going to show you today using a lab paradigm. Another point that I want to make is that I'm going to be focusing on this one paradigm today that really models one type of self-regulation, one teeny little strategy. I'm not equating this strategy with all of emotion regulation or all of self-regulation. There are many different ways that we can attempt to regulate our emotions, but I'm going to be focusing on this one strategy, which is a variant of cognitive reappraisal. Particularly, it's a strategy that involves emotional distancing. The reason that I am going to be focusing on this strategy is that, first off, there's been a lot of work previously published on it, so we have a good sense of what it looks like to deploy this strategy in adults, both in healthy and in clinical samples, at the level of brain and behavior. So if that's a helpful sort of starting point for us as we try to map out the developmental trajectory that gets us to that adult state. But some other reasons for why it's useful include the fact that some of the skills that are often taught in accepted therapies involve using cognitive strategies like this one. And then finally, just for feasibility reasons, this is a strategy that's really pretty easy to teach a participant in a short period of time, and we can implement it both in the lab and in an MRI scanner. So with that being said, how do we do this? In order to assess what happens when somebody experiences an emotion, one way in which my lab attempts to look at this is just by presenting people with a variety of different kinds of emotional stimuli. So in a given experiment, participants may see something like 100 different images, half of which are likely to be emotional in nature or negative in nature, and half of which are likely to be neutral. So just to give you an example, here's a sample neutral image of just two kids sitting in class next to each other, surrounded by classmates. Hopefully this is not super emotionally evocative for you. This should be a neutral stimulus. contrast, if we want to look at what happens when someone's actually experiencing an emotion, we might present them with something a little bit more aversive, such as this child bullying another child. Now, we can compare the way in which people respond to these two different kinds of images to get an assessment of what their emotional reactivity looks like. That is, their kind of bottom-up, knee-jerk, automatic emotional responding. We can also, however, instruct participants about how to respond to the negative images in order to compare what happens when they regulate their emotions, that is, when they use top-down effortful control to change the way they feel, as compared to when they react in a more natural way to aversive stimuli. And the way in which we instruct participants about how to respond is simply with the presentation of an instructional cue. So participants are trained prior to beginning an experiment that when they see the word close, this means that they're supposed to imagine yourself inside of the picture, standing very close to the people you see. So we're not asking them to regulate in any way. This is sort of our baseline of what happens when people do not regulate their emotions and respond to an emotional stimulus. By contrast, when they are presented with the word far, they know that this means imagine yourself being farther away from the people you see. Imagine you are making a movie about them. So we're not explicitly telling them to regulate their emotions or to feel better, but instead we're asking them to adopt a slightly more objective and distanced perspective, with the idea being that this may incidentally cause them to experience less negative affect. So in order to assess whether this is working, whether using this regulatory strategy actually reduces negative affect, the first way in which we can assess this is just by asking people to report on how they're feeling. Now, self-report often gets a bad rap. I love self-report. I know people have different opinions about this, but I really love self-report in this paradigm because I think it's the really sharpest indicator we have of affective experience. The reason I say that is, at least compared to a lot of other options, it's a more precise index of how people are feeling. So there are other tools that I've used in other kinds of paradigms, for example, looking at skin conductance and other kinds of physiological indicators of autonomic reactivity. The issue with using something like that in this paradigm is that if you're asking someone to use an effortful cognitive control strategy to regulate their emotions, you might expect that to increase physiological arousal. At the same time, if you're asking them to use that strategy so that they have less negative affect, that ought to reduce their arousal. And it can be a little bit complex trying to take apart those two components to figure out how to interpret different measures of physiological reactivity. Self-report allows us to bypass that. The other reason I like using self-report is that we can specifically look at something like negative affect rather than overall just how revved up a person is. And finally, it's something that we can use really nicely as converging evidence alongside neuroimaging data to be able to articulate what it is that's changing across age. And that's something that I'll be showing you quite a bit of in the next few slides. So in first focusing just on the behavioral data on self-report, using this paradigm that I've already presented to you, the first question we can start to get at is whether age predicts differences in emotional reactivity or in regulation, or perhaps both. So here is a graph from the first study I ever did looking at this with this paradigm. What you see here are participants' self-reported negative affect values on the y-axis. Larger values indicate more negative affect and they're feeling worse. Lower values indicate they're feeling better. And the first thing we want to compare here is how participants respond when they're engaging in the more emotionally reactive conditions shown in red, as compared to when they actively regulate their emotions shown in blue. Across this age range of 10 to 22-year-olds, we see that participants were capable of using the regulatory strategy to reduce negative affect. So even in our group of 10 to 13-year-olds, so individuals who are kind of transitioning from childhood to adolescence, even in this youngest age group, they are capable of using this regulatory strategy to reduce negative affect to some extent. At the same time, we observed significant age-related improvements in the ability to self-regulate, such that our older participants showed the biggest drops in negative affect, and our younger participants showed the smallest drops in negative affect during regulation. When we look at emotional reactivity, there are no age-related differences here, leading us to tentatively conclude observed at least, that it's really regulation that's changing as a function of age, while reactivity is remaining constant. Now, I want to insert two caveats here to this claim. The first is that I think in everyday life, it's not always so neatly separable, whether we are responding in a reactive or regulated way. It often gets mixed up, such that, especially if you're an adult who's gained many years of regulatory experience, we might start to regulate in more automatic ways, so it can be difficult to tease these things apart. We're specifically using this experimental paradigm for a reason, so that we can dissociate reactivity and regulation, not because we're trying to simulate what people do exactly in everyday life, but to try to identify the underlying mechanisms that give rise to age-related changes in emotional experience. The second caveat I want to point out here is that this is looking at individuals 10 and up. So I made this claim about how 10-year-olds are capable of using this strategy, 6-year-olds not so much. So we have gone down to ages as young as 6, and there's something magical that appears to happen at around the age of 10. Before that, kids are really variable with their ability to use this strategy, at least when it comes to turning down negative affect. I've taught kids as young as 6 to use a strategy like this to reduce food craving very effectively. But there's something about negative affect, and especially learning how to regulate responses to negative social stimuli that's really tough for children. And it's only at around 10 that we start to see them being able to reliably use a strategy like this. So happy to talk about that later if you're curious. But for now, I just want to leave you with the feeling that this is a result that you can believe in, because we've replicated it many times. Here I'm showing you across 3 samples. We've since replicated it in 2 other samples, and now 2 other labs have replicated it as well, suggesting that if there's one thing you believe today, believe in this. I personally always look at everything I collect with a little bit of doubt if it's the first time I see it. The third time, I start to feel a little bit more comfortable. So we've observed now over and over again evidence that regulation improves with age while reactivity remains constant, at least in the context of looking at self-report. We've also started to examine how this might be supported by age-related changes in brain development. So just to back up for a second, before I start telling you about age-related change, I want to focus on what are the suite of brain regions that we might expect to see supporting self-regulation, broadly speaking. So these are the results of a meta-analysis that I was a part of now several years ago, looking across about 50 studies in healthy adults, looking at what brain regions consistently are activated when individuals deploy cognitive regulatory strategies like the one I told you about today, in terms of what brain regions show increases in activity and what brain regions show decreases in activity. Broadly speaking, we observe that in healthy adults, when individuals attempt to cognitively regulate their emotions, they show increased recruitment of a suite of dorsal and lateral prefrontal cortices and posterior parietal cortex, all of which are thought to be involved in a number of domain general cognitive control processes, including selective attention, working memory, and response inhibition. At the same time, we see that engagement of these kinds of strategies seems to be associated with reductions in activity in the amygdala, which, as I'm sure many of you are familiar with, is a subcortical structure that's important for detecting salient information, for forming affective appraisals, and for binding these kinds of information into memory. So what this tells us, or at least suggests to us, in the aggregate, is that when we deploy cognitive strategies, we're using cognitive control to transform the meaning, the affective significance of a given stimulus, by holding it in our minds, by changing it in our working memory, and through these sort of cognitive transformations, we're literally changing the way in which we interpret its meaning. So how might this change across age? Well, we've now observed across a couple of different studies some consistent patterns, such that as individuals get older, I should say actually that in association with age, we see, because these have been cross-sectional studies thus far, we see that older individuals show greater recruitment of lateral prefrontal cortices in the context of self-regulation, and also that age is associated with significant reductions in the amygdala response, specifically when regulating over and above what we see during reactivity. Now, we not only observe these 2 patterns in isolation, we see that they're also related to one another. So specifically, if we look at this relationship between age and the amygdala response and try to see whether it is connected to in any way what we observe in lateral prefrontal cortex, we find that indeed it is, such that greater recruitment of ventrolateral prefrontal cortex appears to mediate the relationship between age and this diminished amygdala response during self-regulation. So what this suggests is that these age-related changes in the amygdala response hinge, at least in part, on a growing ability to recruit lateral prefrontal circuitry that we know is important for selective attention and response inhibition, perhaps suggesting that as individuals get older, they become more adept at identifying regulatory strategies and holding them in mind and being able to use something like this distant cognitive frame so as to change the affective significance of a stimulus. So we've now observed this a couple different times. One thing that I'm not showing you here in the interest of time, but just in case anyone is curious, is that we've also observed that simply recruiting ventrolateral PFC might not be enough to turn down the amygdala response in childhood and adolescence. Indeed, it seems as though we also need to see evidence of connectivity existing between the ventromedial prefrontal cortex and the amygdala. That seems to serve as something of a key that allows these more lateral prefrontal regions to exert their regulatory effects. So that could be an indication of one of two things. One could be that BMPFC amygdala connectivity is a just good index of brain maturation, and maybe that's all that is for why this is important for lateral regions to be able to work their magic. The other possibility is that what we're actually seeing here, this relationship between ventrolateral PFC and the amygdala, is supported by BMPFC as an intermediary, which could make sense from a sort of circuit perspective, that it's really a lateral to medial prefrontal action that's then acting on the amygdala during these kinds of cognitive regulatory efforts. So those are two possibilities that we're continuing to follow up on. So far I've shown you evidence that age is associated with a growing ability to turn down negative affect and to regulate the amygdala response, and that this depends in part on maturing lateral prefrontal circuitry. What I want to talk about now is another way in which we've started to examine the development of self-regulation, which is by not only looking at what people do in the moment when they're self-regulating, but whether there are enduring impacts of those efforts over time. So the way in which we've done this is by, again, having children, adolescents, and young adults complete this same paradigm I showed you before, and then surprising them by telling them they're not getting out of the scanner, but in fact, they're going to sit there and look at some more aversive images for a while. They're usually pretty amenable to this. But anyway, so what we do is we present them with the images that they previously saw in the paradigm that they had seen 20 minutes earlier. And we tell them, you're going to be seeing those same pictures you saw before. Don't have to worry about close or far or any of that. Just look at the pictures. And 2 seconds is a really pretty brief period of time to look at these images. It's not enough time to be actively trying to regulate your emotions. And we did this for a reason. We wanted to examine, just as we had before, amygdala responses during the act of regulation. So this is what the same kind of pattern I showed you before, that as individuals get older, they show this reduced amygdala response in the context of regulation. But we also wanted to look at amygdala responses during this representation period and see whether having previously regulated your emotional response to a stimulus had any enduring impact on the way in which your amygdala responded to that stimulus later on. And here, the age effects that we previously observed are even more magnified, such that for older individuals, we see that having previously regulated your emotional response to something completely changes the way in which your brain responds to it the next time it's presented with that stimulus. Whereas when we look at the children and adolescents, that prior act of regulating doesn't seem to be buying them anything. If anything, there might be a slight trend in adolescence towards this actually increasing amygdala reactivity to the stimuli they previously regulated. So what this suggests to us is that the effects of regulation become more enduring as a function of age. So I think this is really fascinating from a basic science perspective, just because I think it illustrates what a rich range of opportunities may exist for looking at interactions between self-regulation and memory systems. That's something that my lab is currently following up on, some behavioral work in young adults. I think it's also really interesting from a translational perspective, such that if we want to understand what some of the key features are that may distinguish self-regulation between individuals at different points in age, we not only need to look at what's happening in the moment, but how it sticks with them over time. So whenever I present these findings to my friends who are clinicians, this really resonates a lot of times with the feeling that you might make a ton of progress with someone in the context of therapy, feeling like you've really changed their perspective on something, they're seeing things in a whole new light, and then like a week later it's as if that never happened. I think a similar thing might be happening here when we consider what's going on in adolescence, where there might be age-related differences not only in the way in which we regulate our emotions, but also whether or not that sticks with you over time. And so it's, I think, another important layer to keep in mind as we try to unpack age-related differences in these phenomena. So to summarize what I've shown you so far in this first part of the talk, I presented data to suggest that that age is associated with a significant improvement in the ability to self-regulate, as indexed by negative affect and also by responses in the amygdala. I've shown that age-related changes in the amygdala response hinge, at least in part, on growing engagement of lateral prefrontal cortex, and also that age is not only associated with changes in the ability to self-regulate in the moment, but also in how that persists over time. So with that in mind, I now want to switch to talking about some more recent work that my lab has been pursuing, looking at whether we can create quantifiable metrics to identify neural specialization and how that may support the growing ability to self-regulate. So broadly speaking, taking a step back from self-regulation, just thinking about what development is, we can think of development as being characterized by specialization. That is, when I talk about specialization, I really mean two things. One is less variability, and the other is greater efficiency. So I'm always struck when I go to my daughter's daycare to watch the different children, all of whom are pretty much toddlers, how they figure out how to locomote around the room. If you ever watch this, it's really amazing. Because I think sometimes if you haven't spent time around small children, you just think they crawl and they walk. It's not that simple. They do some really weird stuff. So if you watch little kids, some of them will scoot on their butts backwards on the floor. They'll scoot forwards on their butts, realize that's kind of painful. They will crawl on hands and knees. They will bear crawl. There was one girl who would walk on a diagonal like this for like 3 months, and then one day she just stopped. Kids are trying a million different strategies to figure out what works. They're incredibly variable during times of high plasticity because they're trying to see what it is that's going to behoove them in the long run. And as we mature, you start to see this trade-off where flexibility might go down a little bit, but you gain expertise. So variability ought to be decreasing, at least to some extent, but in exchange for that, you ought to see greater efficiency. So we have a good deal of evidence to support this general claim with regards to structural brain data, but only recently have folks started looking at this with regards to brain function. And in the work I'm going to tell you about today, we really wanted to start to think about whether this might be a useful framework to consider the neurodevelopment of self-regulation. So one piece of data that supports the idea that this might not be a totally ludicrous idea is the fact that if you look in adult samples, adults who have undergone a kind of training regimen where they practice self-regulatory strategies like the one I've been telling you about today, you see changes in brain function that are supportive of this idea of greater specialization, such that you'll see that after practicing using a strategy over and over again, they show increasingly focal prefrontal activation during self-regulation, but they achieve their goals quite successfully. So they turn down the amygdala response, they report less negative affect, without needing to recruit all of prefrontal cortex, just the smallest of regions that seems to be really useful. And this has led us to want to examine whether age-related improvements in emotion regulation might be driven by neural specialization. So not necessarily only looking at individuals who have undergone something like an extensive training regimen, but just during natural development. Might this be something that unfolds? And this has been spearheaded primarily by my graduate student, John Wassi-Barrera. So if you have questions or if you don't like it, you can just blame him. This is mentoring in action. All right. So this This work has been done in collaboration with Kate McLaughlin at Harvard University, and in looking at a sample of 70 typically developing youth ranging in age from 8 to 17 years, we've really been initially focusing our efforts within regions of interest that we have strong existing hypotheses should be involved in self-regulation. So we've looked at several different regions of the brain. I'm going to be focusing today just on ventrolateral prefrontal cortex because this is a region we know is really important for self-regulation in adults, and we also have prior data to suggest that there are significant age-related changes with regards to just gross activation of the LPFC across age. So using the same paradigm that I had told you about earlier, it's just a teeny bit different with regards to the baseline reactivity condition, but otherwise it's pretty much the same. We wanted to look in this initial study at two different measures that we thought might be reflective of specialization. The first is spatial specialization, with the idea here being that if a brain region is showing spatial specialization, we ought to see a very specific subset of voxels being activated in the context of self-regulation, and that this ought to track with either age or ability, perhaps both. We went into this really with kind of exploratory framework. The other type of specialization we wanted to look at was temporal, with the idea here that, as I was saying earlier, we might think of development as being characterized by less variability. And so what we did here was wanting to see whether there might be less variable trial-to-trial activation with regards to a given brain region's pattern of engagement. So to turn first to spatial specialization, John had a really creative idea for how to look at this, and it came from a somewhat surprising place. So he reads very widely, and he found in combing the literature that in macroeconomics, one way in which people estimate income inequality is with something called a Gini coefficient. And a Gini coefficient basically is reflective of how wealth is distributed in a society. Are there very few people who hold a lot of wealth, or is it more evenly distributed? And what we reasoned was that we could apply a similar framework, and it had been done just in one study that we could find so far in adults, to understanding not necessarily income distribution, but activation distribution across a given brain region. So what we argue here is that if a smaller number of voxels are accounting for more of the activation within a region of interest, this might be a proxy for specialization. So in looking at this, we did find preliminary evidence to suggest this might be the case, such that those individuals who showed greater specialization also tended to report greater drops in negative affect during self-regulation. When we turned next to looking at temporal specialization, here we were looking at variability, taking single trial estimates of the signal in the same region of interest, and looking just at how variable these estimates were from trial to trial, with the idea that less variability ought to be indicative of greater specialization. And we observed here that less variability, i.e. greater specialization, was associated with age. So here we're tentatively concluding that age and regulation ability might be associated with specialization, albeit in somewhat different ways. So this was the first study we did this in. We're currently in the midst of work to try to replicate this in other samples. But preliminarily, I think that there's something to consider with regards to why we're seeing these different kinds of specialization tracking with age versus ability. So with regards to temporal specialization, we saw this general pattern of less variability across several regions in the brain. This might suggest that when it comes to temporal specialization, and there being these changes in variability, this might be a more general metric of brain maturation. Whereas what we're seeing here with spatial specialization might be specifically reflective of experience. That's how I'm interpreting it, at least. That it might be something about this specific skill in this specific region for why we're not seeing this everywhere, but rather only within regions that we think are most important for something like self-regulation. Such that as an individual actively is attempting to self-regulate in their everyday lives, they're gaining more and more experience, they're gaining more and more ability, that this might be tracking with this spatial specialization, which is somewhat akin to what was observed in that one training study in adults. So I'll return to these possibilities at the end if you want to discuss that further then, but for now, I want to tell you about one other way that we started to look at neural specialization. So in the same sample of children and adolescents, we started to look not only within a given brain region at how specialization might occur, but also across the network. So in taking those the same data that I showed you earlier from that meta-analysis of looking at emotion regulation in adults, we took that meta-analysis and just broke up the different clusters into subclusters, which are depicted here as nodes. And we've looked at network specialization in 3 different ways. I'm going to be presenting just one of them today, which is asking how distinct are connectivity properties during regulation versus reactivity. So the idea here is if the brain is getting really specialized for something like regulation, it ought to demonstrate a unique multivariate signature with regards to connectivity in that context that is not demonstrable in other contexts. So put another way, if we look at the pattern of connectivity across the different nodes in this network, they ought to appear different in the context of reactivity versus regulation. And the more dissimilar these 2 different correlation matrices are, might be reflective of greater ability. And indeed, we have found preliminarily that this is the case, that greater connectivity dissimilarity is associated with greater regulatory ability. So leading us to suggest that emotion regulation network connectivity tracks with regulatory ability. We are seeing some other network properties that track with age, but it doesn't seem to be this dissimilarity assessment. It seems as though there are some other features of how the network clusters apart into different components that does track with age, but with regards to ability, it's really this dissimilarity matrix that seems to be doing something. So to summarize what I showed you in the second part of the talk, I presented data suggesting that the extent to which youth recruit smaller portions of lateral prefrontal cortex more robustly tracks with their self-regulatory ability. And I've also suggested that at the network level, we observe another layer of specialization, such that the extent to which individuals demonstrate distinct patterns of connectivity in the context of regulation versus reactivity is also reflective of this greater regulatory ability. And we're thinking about both of these markers potentially as indices of specialization and of ability. So I wanted to conclude with some future directions, both to tell you where we're going and also hopefully to spur some interesting conversation during Q&A. So one question that continues to be on my mind is whether age-related differences in self-regulation are biologically locked, so to speak, or dependent on experience. So when I talk about things like the fact that children under 10 are terrible at using this strategy, is that because their brains are simply not ready to try to engage a strategy like this, and no amount of experience would change that? Or is it just that there's some critical age at which you've massed enough experience with regards to self-regulation that you suddenly are capable of using it? So I think that one way we can do this is by testing through manipulating experience. So I would love to, at some point, we've had several attempts of starting this, but to work hand in hand with a school to try and see whether children practicing a strategy like this over and over again has both demonstrable effects on their performance in a lab-based task, but more importantly, whether that generalizes into their everyday lives, to see really what the role of experience is in developing these different skills. Another very different way in which we're examining the role of experience with regards to the acquisition of self-regulation is by comparing children who have had really different early experiences. So specifically right now, my lab is conducting work in a large group of children who are actually now adolescents who were in orphanage care for the first couple of years of life before being adopted. And we're examining how those early social experiences, or lack thereof, compare, or how they impact long-term development of these systems compared to children who were raised in more typical rearing environments. Another question that we think about a lot is how different forms of emotion regulation co-develop, or whether they do co-develop. So today I've really just stuck with this one paradigm, looking at one regulatory strategy, but I don't think any of us rely exclusively on just one way to regulate our emotions. That wouldn't be super adaptive. It's a lot of effort to use this cognitive distancing strategy. I personally only like to whip it out when I need to. There's a lot of things that are easier to do. So rather than having to sit there and transform the meaning of cookies, I can just not buy cookies and take that out of the equation. There's distraction, there's social support, there are a million different things we can do. And one of the challenges in the field of emotion regulation is that many different researchers pursue many different strategies, but it's really tough from a lab-based experimental context to look at whether the same individual is capable of deploying many different kinds of strategies. But that's exactly something that we think is really valuable to do, to see, for example, whether you need to be able to label your emotions before you can reinterpret them. I think that there's some really basic questions that need to be addressed here in order to really understand what the developmental trajectory of emotion regulation is in this broader context. So with regards to the latter part of my talk, looking at neural specialization, we're continuing to ask whether that's what we're really studying or not, and doing so in a couple ways. One is by leveraging longitudinal data in youth, so just youth who have completed this paradigm that I've been telling you about multiple times, and seeing whether they show changes in these different metrics of specialization, but also looking in adults who have completed a training regimen and seeing whether they show changes as well. And then finally, we're looking at not only the aspects of network specialization I told you about today, so here I presented data looking at connectivity dissimilarity, but we're looking at other features of networks and how that may also map onto age and regulatory ability. So specifically looking at whether, first of all, this is specific to if this is specific to this one brain network that we might expect for these patterns to matter, or what it looks like in other brain networks as well. But we're also testing other network characteristics, including the sort of subcomponents of a network, how many different communities it might break into, and also modularity, or the extent to which those different communities are communicating with one another. So those are just a few of the ways in which we're currently pursuing this work. I want to conclude by acknowledging the many folks who contributed to this work over time, and also, of course, thank you for your time, and open up for questions.
Yeah, I was wondering if the connectivity patterns of people who could successfully regulate were more similar across individuals than those who could not.
You know, it's a good question. Our focus was so much on within individual differences. We could examine that, I guess, in more detail about whether they sort of correlating them to each other, or by broken up by skill set, and see if there's more of a canonical pattern to being a good regulator. One thing we have done is try to do some predictive analyses of whether or not we can predict someone's regulatory success, and we're getting there. Yeah, so it does seem like we can predict it to some extent, but not necessarily based on, we haven't tried it seeing if it's because there's one pattern that they're exhibiting, so much as we know that we can predict it based on other network features. That's a good question. Yeah?
Very nice talk. I'm just curious how this cognitive distancing strategy we sort of imagining as a movie director.
Yeah.
Does that sort of help you suppress the emotion or distract you from sort of reacting to the stimulus in an active way? Or another way of framing the question is,
What's it doing?
Maybe the default state, like the baseline,
Yeah.
is the distant one, and then they're not regulating. The difference is when you ask them to go close, they're sort of putting themselves into the situation as like a friend or
Yeah.
protecting yourself and sort of.
So I don't think it's the default state, because we've also done studies where we don't ask them to do anything, and that's pretty different from what we see during this distant state. I don't think also that it's necessarily distracting them, although probably all these things exist on a continuum, these different types of components of regulatory strategies. So we're very careful to make sure that to tell them to focus on it, but just to take a step back. The reason for that is that we want them to continue to engage with the stimulus, but just to change kind of how impactful it is on them directly. I will say that this is a kind of interesting strategy. So part of why I started using this as opposed to more traditional reappraisal strategies was where you're asking them to say something like, if you see a child bullying another child, maybe they're just in a movie, maybe they're playing around. Was first of all, I found it really uncomfortable to train people to disbelieve, but also kids were terrible at it. They were just like, no, that's what's happening. And so I found that using this strategy of more this distancing strategy worked a lot more effectively. So we've done a couple of different flavors of it, including things like focus on the colors of the picture. So they're still looking at the same stimulus, but focusing on something different. When it comes to something like looking at unhealthy food, thinking about the cookie as being circular rather than thinking about how it tastes. So you're still thinking about the stimulus, but it's sort of changing what you're drawing your attention to and what your perspective is. Yeah?
Have you seen any gender differences across development?
No, kind of. It depends. So nothing I trust, honestly. That's the truth. But we haven't tried hard enough. I think to do that well, we would need a big sample in a more limited age range, but it would have to be huge. I mean, there have been a couple published studies suggesting there might be gender differences in adults, but even those, I think they're underpowered to really be able to say that strongly. And the reason I wouldn't trust it too much in our data is that given that our girls tend to develop a little bit faster, how much I would attribute it to that versus gender, I just wouldn't trust it without a much bigger sample than anything we've had. Andrew?
Jen, when you talk about experience, you're talking mostly about regulatory experience.
Yeah.
So can you model or think about the sensory reactivity experience that recognize some of this bottom line? So is there a sweet spot of reactivity that's important to experience?
Yeah, that's a really good question. I mean, I think that, so I don't know if this is where you're going with it, but from my mindset, it's important for youth to experience enough stress to have the opportunity to try and regulate. And so we haven't exactly looked at it from a reactivity framework, but we have been collecting some data on just daily stressors and sort of questionnaires to look at whether your kind of life stress might have more of a linear or quadratic effect on some of these processes. And I think that's really an open-ended question. We have this sort of extreme case of kids who experience this profound early life stressor, where the data so far suggests they are not good at using this strategy. But we haven't looked at more normative variations in terms of how much stress individuals experience and at what ages is that important. I think it's a great question.
So, sorry, I quick follow-up to that. So the reactivity and self-regulation. Reactivity is almost state constant.
Yeah.
Why is
Why? I think
so with experience, could you damp down the reactivity?
so. There's a couple of interesting things here. I think in everyday life, these are not as neatly dissociable as they are in a laboratory context. One piece I think that speaks to this is interesting. A friend of mine who's done a lot of work with training studies in adults, where he teaches people this strategy. They don't report that this strategy is doing anything. They start to just report less stress in their everyday life. And when you test them on this paradigm, after they've practiced it over and over again, they start reporting less negative affect both with reactivity and regulation, suggesting that it starts to become more implicit if it's something you practice over and over again, and that it can potentially impact just global affective responding. I think that when with a paradigm like this, when we're introducing it to people in this sort of novel context, we can kind of pull them apart, but it's really just to get a mechanism. Yeah?
I was wondering, could you elaborate a little bit on the relationship between emotion regulation development and the development of cognitive control?
Yeah.
Or is
It's so understudied. It's this weird thing where often people just kind of, sometimes they conflate them, sometimes they act like they're unrelated. So there are a few studies that have suggested this kind of regulatory strategy is positively associated with performance on some cognitive tasks. That's not shocking. They're not 100% the same, but they are related. What's tough is that when you look at them in development, both correlate with age, so it can be a little tough to figure them out. My read of the literature is that cognitive control seems to emerge a little bit before the ability to deploy strategies like this. But that kind of makes sense that anytime you layer some kind of emotional intensity onto something, it should take a little longer. Similarly, we've seen sort of the ability to deploy this kind of strategy is dependent on the stimulus being used. So like I said, kids are better able to use this strategy when they're in food craving at a younger age than when it comes to negative stimuli. Negative non-social stimuli, they're better at regulating sooner than negative social stimuli. So if you think of cognitive control as sort of some of the underlying support structures for that, it makes sense that that might emerge a little bit earlier. But yeah, I think they are related, and it's something that we're looking at to see how more interrelated they are.
One last question. speak afterwards.
I wasn't sure if you were pointing to someone.
I think she raised her hand.
I was just going to ask, it seems like most of this research has been done in community samples, and if you could just talk a bit about, if you use these paradigms in clinical populations and what that research looks like.
Not in pediatric clinical populations. When I was a graduate student, I did some work in adults with borderline personality disorder, and as anyone who's been a grad student or currently is, can attest to, sometimes free data aren't free and that you don't get to design the study. So in that sample I worked with, we didn't have a control group. So it was just looking at variations among individuals with borderline personality disorder. So one finding we had there was that the extent to which individuals recruited ventrolateral prefrontal cortex tracked with self-reported difficulties in emotion regulation in everyday life, whereas differences in their ability to turn down the amygdala response were more closely associated with affective lability in the context of BPD. There have been other individuals who have used this kind of paradigm in other clinical samples. So actually that study I was talking about where we looked at the enduring effects of self-regulation, that was modeled after some work that Suzanne Erck has done in depressed samples, where they similarly find that individuals who have depression are capable of turning down their amygdala response in the moment, but it doesn't stick with them over time. So there's definitely been work using this kind of paradigm, but I haven't done it in a clinical pediatric sample.
We have 5 minutes, so I'm going to allow 2 questions.
Hi.
Have you were looking at the inhibition in the amygdala response, have you looked to see if there's shift to other subcortical areas, like any change in the mental state?
Oh, like if regulation maybe increases positive?
Right, are they able to shift to different emotions?
Yeah, so other people have done that looking at slight variants of strategies. So for example, if you use a strategy that involves trying to find the positive features of a negative stimulus, you do start to see more recruitment of striatal regions. For this strategy, because it's really almost more of a dulling effect, I think, that we're seeing, where they're distancing themselves, we haven't seen that. The one way in which we've ever seen striatal responses was in the context of regulating food craving, where we see diminished striatal responses. That's a good question.
So for the connectivity dissimilarity analysis, I'm just curious, sort of, in the emotional regulation network that you defined, there were a lot of clusters of nodes that were really close together anatomically, and it could be the case that the spatial specialization findings, where you get sort of more specific voxel patterns, function of age, could also be driving that effect. Like, would you look at sort of the long distance connections versus the local connections? Because if an area is all kind of doing the same thing, specialization would just be high connectivity also.
Yeah, it's a good question. So, I think one way in which we can start to look at that is partially actually by looking at whether these different metrics of specialization are associated with one another in any way. But also, we have looked at, if memory serves, and I guess John, because he's there, I think we did look at controlling for the distance between the nodes at one point, or we talked about it, but I can't remember if we ultimately found how much relevant that was.
We talked about it.
I know we did. That's where we left.
Yeah. Sparse with fewer nodes.
Yeah. That's a good point. I think this is one of the questions that we sort of struggle with is how to quantify the just proximity and wanting to take that into account versus the sort of general structure. And so we have talked about it and toyed around with a few different possibilities, but I think that's a good question, so that we can pursue before we actually write up the paper.
Okay, so it's a very nice discussion. So let's thank Jennifer again.